So in this session, we're going to be talking about natural killer or NK cells. You'll notice I put another picture of a cat I have. His name is Yoda. He's a Cornish Rex. He's just a weird looking cat. And be more than happy to talk about him. Okay, so we're going to talk about natural killer cells, NK cells. So we're going to talk about what NK cells are, what is the difference in killing, how do they kill, how do they recognize cells in comparison to T cells. We're going to talk about the functions of NK cells, explain the mechanisms through which they recognize normal and target cells. We will talk about some of the different receptors involved in NK cell activation, and we'll explain the mechanisms involved in ADCC. And then we're going to discuss not only NK cells, NKT cells, and Lac cells. And what are the differences between those? So what are NK cells? Now, NK cells are actually a lymphocyte. They develop in the bone marrow from lymphoid progenitor cells. It is not a B cell, nor is it a T cell. And in fact, they used to be called large granular lymphocytes because they had a bigger size and had lots of granules in the cytoplasm. So they're larger than B cells and T cells. They have lots of granules in their cytoplasm. They're cytotoxic. And so when you look at them under a microscope, they're large and granular. Now, they can kill cells without prior immunization or activation. They're more in the innate immunity rather than adaptive immunity. So they don't have to have seen those target cells, the viruses, tumor antigens, etc., before they have the capacity to kill those cells. Now, the granules in these cells, the cytotoxic granules, are very similar to the ones that are found in cytotoxic T cells. And they kill cells through apoptosis, just like cytotoxic T cells. Again, they can kill virally infected cells, tumor cells, and they do not have antigen specificity. So they don't recognize a viral antigen or a cancer antigen like cytotoxic T cells will do. These will recognize a cell when they look like they're sick, and we'll go through that in a minute. These cells can also mediate antibody-dependent cellular cytotoxicity, and I talked about this already. Another name for this is ADCC. And basically what this is, just to refresh your memory, is that an antibody molecule, an IgG, will bind to the surface of a target cell. It will recognize a viral antigen or a tumor antigen. And the natural killer cells is one of the cells that can recognize this antibody on the surface, and because it has an Fc receptor for IgG. And so when it recognizes this, then it could kill that cell. They also can produce cytokines after activation, and gamma interferon is one of the major ones that it can produce, but there are other cytokines it can produce. And so these cytokines is, again, they're peptide hormones, and so they can influence immune responses and activate macrophages or promote Th1 cells. So natural killer cells are an early part of the response to viral infections. The main thing is they can help control virus replication early on. And you can see here in this graph, time after viral infection in days. And it's monitoring viral titer, that's that yellow thing in the back. And then you see first there will be a production of interferon alpha and beta and TNF alpha and IL-12. A lot of those are from those infected cells. Okay? And those help stimulate natural killer cells, and natural killer cells will come in and hopefully control the virus infection. So you can see where in this idealized pattern, the virus infection, when the natural killer cells come up, it doesn't eliminate them, but it keeps it from growing very high. And then eventually it gives it time for the cytotoxic T cells, the adaptive immune cell response to generate. And those T cells, cytotoxic T cells, can come in and finish off the job and kill the virus kill the viruses. So natural killer cells early on can help control the virus replication before it gets out of hand. Virus is eliminated by the cytotoxic T cells. And if you don't have natural killer cells, virus levels can become higher, and it may be so high and so out of control that it's too late when the cytotoxic T cells come in. So the natural killer cells give you some time before the infection gets out of control. The interferon alpha, beta, and IL-12 help activate natural killer cells, and they also help increase their killing capacity. So they activate them, and that results in their increasing their ability to kill infected cells. IL-12 and TNF-alpha can also act synergistically to stimulate interferon gamma production from natural killer cells, and that's where natural killer cells can be producing cytokines that can activate other cells, such as macrophages. Now, how does a natural killer cell recognize a target? Well, natural killer cell killing depends on a balance between activating and inhibitory signals. And it's called a missing self. They measure certain molecules on the surface, and when those disappear, they recognize, oh, something's wrong with this cell, let's just kill it. So the natural killer cell has several different activating receptors that recognize common carbohydrate ligands on the surface of the cell. Natural killer cells are prevented from killing any cells when another cell of receptors recognize MHC class I molecules. Again, in humans, that's HLA-A, HLA-B, HLA-C. And so when that molecule, that's receptor on the surface of natural killer cells, recognize the class I molecules, that triggers an inhibitory signal and prevents that activating receptor from activating the cell. If MHC 1 molecules disappear, that inhibitory signal is lost. And when that occurs, cells will get killed. And the reduction of MHC molecules can occur with viral infections or on cancer cells, where in an effort to avoid killing by cytotoxic T cells. So the idea is the virus shuts down MHC class I. The virus has evolved knowing that MHC class I is something that the immune system, the CD8 cells, can recognize. So they shut it down in the effort to avoid being killed and removed by cytotoxic T cells. Now I'll give you a little more of a graphic about this whole thing. So NK cells recognize normal cells expressing MHC class I. So NK cells have an activating receptor that recognizes a carbohydrate on the surface of a normal cell. As you can see, that green little activating receptor. They also have an inhibitory receptor. That inhibitory receptor binds to MHC class I. So what happens there is in that inhibitory receptor binds to MHC class I, it dampens, inhibits activation through the activating receptor. So the natural killer cell will not kill a normal cell. However, if you have an altered or an absent MHC class I molecule on the surface, then the inhibitory receptor can't recognize that. And so therefore the activating receptor, the green receptor there, can send a positive signal to the natural killer cell, and that natural killer cell releases the granules and resulting in the induction of apoptosis in the target cell. Okay, this is called the missing self hypothesis. That means you're missing the MHC class 1, which would be self, or it's an altered MHC class 1, which would be self. So you're missing that, and that results in killing. Okay? If you have self, that inhibits the activation, and the natural killer cell won't kill the cell. So this is what I mean. If a virus infection or a cancer cell looks sick, what I mean by that is it's missing the MHC class 1 molecule on the surface. So tumors lose MHC class 1 to escape immune surveillance. Okay, so basically this is a series of studies that were done to examine this. So on the far left, you see a tumor cell displaying a novel antigen, and you inoculate these into a normal mice or nude mice. Now, nude mice, what they do is they have no T cells. They lack a thymus. And so when you start looking at the cytotoxic T cells, or the percentage killed, you can see that most of the killing is done by cytotoxic T cells and a little bit by natural killer cells. And you can see in the tumor mass that nude mice, the tumor mass goes straight up, and in normal mice, at a time when cytotoxic T cells develop, the tumor mass disappears. Now, if you take that tumor cell and you lose MHC class 1, and you make a variant to that, you will see that normal mice, that tumor mass will get out of control. But in the nude mice, natural killer cells that are present in the nude mice will take over the killing. And it's because they recognize the loss of MHC class 1. And in the last one on the right, if you transfect with take that variant where you've lost the class 1 and put back in the MHC gene, it goes back to being normal. And this basically shows that whole idea that natural killer cells recognize the loss of MHC and will be an effective killer by itself. Where in normal situations, cytotoxic T cells may be more effective in finally controlling the infection. So the nude mice lack a thymus. They have no T cells, and they actually have higher levels of natural killer cells, which is why you see the pattern we've shown you. Again, missing self is the loss of MHC class 1, and that results in the natural killer cell killing that cell. Now there is a number of NK cell receptors. So you have inhibitory and activating receptors. And the inhibitory receptors recognize MHC class 1. And you can see there's NKG2A/B, CD94 heterodimer, the KIR, a couple KIR ones that are all inhibitory. And then you have some activating ones, same sort of pattern. I don't expect you to know all those receptors. You should just recognize that there are inhibitory receptors that recognize MHC. There's multiple ones of them. You don't need to know KIR2DL is one. You just need to recognize that and activate. And there's activating receptors that recognize a ligand on the surface of the target cell, and that will stimulate. If not inhibited, will result in stimulation of the natural killer cells and killing. The other thing that these natural killer cells have is CD16, and that's that Fc gamma receptor. Okay? So the natural killer cells won't have T cell markers on them, B cell markers on them, but they'll have CD16. And they'll have some other cytotoxicity receptors. So they have lots and lots of different receptors on the surface to help them do their job. Now there's a cell type called lymphokine activated killer cells, or Lac cells. And this cell type is produced to help combat cancer. So basically what you do is you take a cancer patient's cells from the blood, or if they really want to get fancy, they'll take them from around the tumors, and they will stimulate them, grow them in the presence of interleukin-2. And they'll do this for several days to expand those cells, and then they'll eventually reperfuse them back into the patient. It has been shown, this type of treatment has been shown to be successful in treatment of some patients, but not all. So again, you take their cells, you activate the heck out of them with interleukin-2, and reperfuse them back into the patient. And it has been shown to be a successful treatment in some patients. Now, Lac cells are primarily IL-2 activated natural killer cells. Again, Lac cells are primarily interleukin-2 activated natural killer cells. And so what this growing in the presence of interleukin-2 does, it increases the synthesis of perforin and granzymes, which, as you remember, they are key components in the cytotoxic granules. Remember, perforin forms the pores. When it gets released, it forms pores that allows the granzymes to get into the cytoplasm of the target cell, and that results in activation of the caspase pathways that lead to apoptosis. It also increases the killing capacity, including against tumor cells previously shown to be resistant to natural killer cells. So something occurs with the stimulation of interleukin-2, where this increased synthesis of perforin, etc., whatever else is going on, has increased the killing capacity, and it'll even kill cells that were resistant to normal natural killer cells. So these are Lac cells. There's another cell population called natural killer T cells, or NKT cells. They express the NK cell markers, such as the Fc gamma receptor, but they also express CD3 on the surface. So they look like a T cell. So if you remember, CD3 is part of the T cell receptor complex. Now, these cells are able to recognize glycolipid antigens from bacteria. And these antigens are presented on a molecule called CD1. It looks very similar to class 1 molecules, but it isn't a class 1 molecule. So it's a glycolipid antigen being presented by CD1 that these cells recognize. These cells have a regulatory function. They secrete large amounts of cytokines. They include interleukin-4 or gamma interferon. These cytokines can help polarize T helper cell subset responses. So, for example, with patients with severe asthma, ulcerative colitis, they can have high levels of NKT cells, And these NKT cells produce IL-4 and IL-13 that promote Th2 responses. So asthma and ulcerative colitis are Th2 cell mediated problems. Okay? So NKT cells can play a role in promoting those harmful responses in this case. In hepatitis C infections, interferon gamma from NKT cells can promote Th1 responses that will help combat hepatitis C infections. So they, through the production of cytokines, they can help steer what type of immune response, sometimes beneficial, sometimes harmful. They can also stimulate cytotoxic T cells, natural killer cells, and dendritic cells. So they typically don't have a cytotoxic function, but they typically have some sort of regulatory function that helps guide the immune system and handle whatever problems it's dealing with.